Okay, this is William Neal with the local group in Biology of Fishes here on September the 11th, 2012. And probably like you, I'm remembering another September the 11th, 11 years ago. So, that's on everybody's mind, I guess, today. And also on our mind, though, a happier note, Allison just told me in the hall that today's her 18th, right? No.
That's right.
Happy birthday, Allison. And I'm pleased to start with a photograph that Ray Kemps sent. We were talking last time about density differences in water masses and how that sometimes results in strange ecology, strange hydrology. And here's an example from Ray's work, or a picture that he sent me, Lake Amistad down on the border. And I guess on the lower left there is the combined flows of the Rio Grande and the Pecos. Wait a minute, is that right?
Yeah.
And then upper right is the lake itself. And actually the lower is more turbid, and this is bluer. The color balance is not very good on the projector here. So this is a blue, clear water and mass, and down here is the turbid water mass. And the two together, Ray explains, push and finally plunge down to the depth at which their density makes them neutrally buoyant. And that's called the plunge point. And he says all kinds of interesting ecologies going on there at that location where there's a lot of mixing, a lot of turbulence. So it's a good example. I looked on the web to see if I could find a picture, an aerial photo of the point of confluence between the Buffalo, a warm water river, and the White River, a cold tailwater, to illustrate another kind of situation where density differences result in separation of water masses or distinct, edges. But I couldn't find the picture. I could find the picture of the confluence, but I couldn't see enough to make sense of it. So I thank you for sending us that, Ray, and I'm sure Ray would be happy to talk about this with anybody in the group that would like to follow up. Speaking of following up, I'd like to do some. I keep, my wife says I'm kind of like a kitty with a fish head. Once I get, interested in something, I can't leave it alone. Just keep worrying it. So I'm still worrying the idea of these data that I showed you. If I can get down to that point in my slide there. There we go. Let me go to here and say that, there's that set of data of DO versus time of day. And, I did some work on this. I took a transient state version of the model that I showed you on DO and CO2 and pH, and I worked on that a bit and asked myself the question, can I explore the hypothesis that Paul Kaysen suggested, which is that maybe it's got to do with aeration, which occurs via paddle wheels. Get my mouse back here in the wee hours before dawn. And so I tried, and I'm not going to show you that. I generated about a half-hour AVI on the work that's posted on the archive site, and I'll show you that in a bit, show you where the links are. You can go and play that back for yourself, but all I'll do is show you the summary statement. This was the best I could do, Paul, in terms of trying to generate the lower part of that envelope, and you can see that I was unable to get the minimum over here where it looks like it is in the data. So it could still be that the paddle wheel aerator effect, keeping DO artificially high when DO is low early in the morning, might delay the onset of the rise that is occasioned by the new day and new photosynthetic activity, but I couldn't do it. I could do it. I could actually generate a minimum that was shifted 2 hours to the right And theirs is my best attempt to fit the central tendency of the time series. And I'm pretty happy with that fit. But I got to tell you, as I said in my presentation, it's kind of heavy-handed in that what I had to do was invoke a delay in photosynthetic production of oxygen for 2 hours. In other words, the argument being that when the sun is low on the horizon at dawn, light penetration is insufficient to really stimulate the phytoplankton to go at it until 2 hours after, to about 8 o'clock. 6 o'clock is about daylight at this time of year. So you're welcome to play that back, and in fact, I hope you will. You're not required to. Well, let's say this. If you're a graduate student in 617, or if you're Ray, you are required to. But the undergraduates, I put this out as lagniappe, to use a good word from Louisiana, a little something extra, a little bonus. And I hope that you will avail yourself of these things that I show you. But you know, it's not a requirement. I want to go to the archive and show you where it is. And where it is right here opposite September 6th, which was the recording. And by the way, I got to tell you that, you know, I played that dude back and I found so many misstatements and wrong, you know, saying up when I meant down and things like that, that I redid it. I didn't redo it in its entirety. I did cut out some pieces about a half hour in where I was not saying things as clearly as I wanted to. And so if you've looked at that September 6th, if you're at a distance or if you're here locally for that matter and you wanted to play back something, I hope you'll re-download it if you've done so. If you downloaded it the first time before about Saturday dark, because I think I posted the revision Saturday evening. And I had I checked I usually use file size to identify the different variants of a file when I upload stuff, and I had it written down, but I didn't bring it downstairs with me, so I don't have it with me. I think it's about 15 megs, and it's a lot shorter than the original, and I don't know why that is. I think it's got to do with the fact that the original was captured while the internet was open, and I recaptured it and integrated some little pieces correction-wise, and that greatly shortened the file to 15 megs for an hour and 10 minutes, which is pretty remarkable. Anyhow, there's the transient state version of the nominal model that I talk about in the AVI that's posted there. About a half hour's worth. I hope you'll take a look. What else? The server was down. This server yesterday, some of the late afternoon. For anybody at a distance that got frustrated, my apologies. Hardy let me know that he couldn't get it to connect, and I got it connect, and I got off campus and couldn't get it to connect. It may have had something to do with going through the firewall. But anyhow, I contacted our IT people, and I don't know if we fixed it or if it just fixed itself, but you know, it came back the very next day, or the very next hour, so to speak. Yeah.
You know, sorry. Going back to the old slide,
yeah.
I was just thinking of what the graph again, and maybe it has to do with what Valdemar just mentioned the other day about the sampling, the planktonic population. Like I remember in a shrimp farm as an intern, there were some ponds that have really heavy, some type of mussel infestation, and no matter the aeration or whatnot, those ponds, the DL level, even during the peak of the day, it never went above 5 or 6 milligrams.
Okay, let me see if I can paraphrase what Alejandro just said, and that had to do with heavy production ponds and a lot of activity keeping DO way down, and that's always the case. But here, what we're talking about is the timing of the diel cycle, and that's what we're trying to get at. We need to set that aside now and finally get away from environment and move on and talk about feeding. But we can continue to talk together on this as much as anybody wants to. So the server was down, but the server's back and apparently working okay. And I'm trying to think if there was anything else that, well, I guess I should say something about our schedule of events. Lab is coming up this afternoon again. This will be the second session of Lab 1. And I will be around, and I'm sure Donovan will be here in the room locally to work with folks that need help to get going with this lab. But you should be prepared to do it on your own with the help of these help files. That's the design. I do have a little presentation I plan to do at the very beginning of the help session this afternoon for local people, and I'll capture that for people at a distance. And what I want to talk about is the application of the password that I emailed all of you, which allows you to open password-protected files, those that are marked with a trailing plus sign. And I'm going to go through that process with you here to make sure everybody is on board with that. I also want to talk about Stella and getting a copy that will work better for you longer than... We're ready to do that. We're ready to get into Stella. So I think that's the front end, the forward that I needed to do. I hate to waste the time doing it, but things have to be said, and they have to be said on the record so that everybody gets the word. I talk to you in the hall, but I don't talk to Hardy in the hall. I'm trying to think of other people that I don't talk to, people who are distant from here. So I want to move on now and talk about foods and feeding in fishes. You've already got the handout locally, the 3 slide per page. The handout is posted on the web for people at a distance. And, we've sort of laid the groundwork. We've got the environment in which fishes have to operate pretty well described, I guess, although there's always some components of that description that are going to be insufficient and deficient. And one of those is the distribution of available forage for fishes. So I want to talk here about what fish eat, and we already know something about how much they eat from Lab 1. So I want to deal with the acquisition of energy and nutrients by fish in sort of a generalized way. It's obviously impossible to deal with it in great detail. But by way of generality, that feeding for success idea at the beginning there is important to me, and I want to sort of say my philosophy and let you kind of weigh it as we go along. And the philosophy is that fish, as individual organisms, just as you and I, have to deal with the world around them. And that means they have to win their resources, and they've evolved to do that very successfully. But individuals aren't equally successful, and that gives rise to competition, intra-specifically or within species as well as among species. And so, it all comes down to this game of trying to acquire more than our individual share of resources, which we then ultimately squander, as we die, and the energy and materials, pass on to something else, maybe just to heat. Sure, we produce gametes, but the actual energy and mass involved in the gametes in most forms over their lifetimes is not a very significant fraction of their total energy and biomass that they've transacted over their lives. So, I think it's easy to get focused on evolutionary success, species success, and I'd be the first to agree that all that really counts from the standpoint of the genome is passing on the genes. But, to get to that point, either humans or fish or any other organism has to win the resource competition battle first before they get to the point where they're mature and can actually produce offspring. So fitness has a lot to do with that whole operation. And we're going to take the focus here in biology of fishes of the individual, because it's really, as I explained at the very beginning, an ecology course, not a synecology course, and not an evolutionary biology course. So I want to talk first about the myriad things that fish eat, and the habits, as we use the word, that fish have in feeding. You know, it's not really a habit; it's more of an instinct, I'd say, a mandate. But we use the word feeding habits to describe the selection of foods that are consumed, and we measure that often by looking in their stomachs to see what they ate. So that's the commonest way of actually looking at feeding habits, is by examination of stomach contents. But it's a lot more fun to actually watch fish feed. You know, it gives you an excuse to get the scuba gear out and get down on the coral reef and, you know, have fun. Or stand above the clear waters of a trout stream and watch as those brook trout come out from behind the boulder and seize that item that comes washing downstream. But, you know, the stomach contents analysis to determine feeding habits, that's kind of I'd say the most common method. And we're going to have some things to say about what you can infer from looking at those kinds of results. You know, if the animal that you're dealing with is coming out of a museum jar, or if it's a fossil in a rock, then you're not going to be very successful either in direct observation or stomach contents. I mean, it might be possible to see preserved fossilized stomach contents, but normally inferences have to be drawn by looking at the anatomy of the preserved specimen. For example, mouth position. And I got some crude drawings here that actually turned out to be kind of artistic, I think, don't you? You know, examining the feeding habits, quote, as a function of where the mouth, how the mouth is oriented. And, you know, if you have a supra-terminal up-looking mouth, up-looking head, kind of like that thing that sort of looks like a tarpon or something there, then probably you're focused on things above you and your surface feeding organism. And similarly, if you're like this thing that looks like, I think it kind of looks like a hogsucker in the North American genus Catastomus, or family Catastomidae. Hypentilium, I think, is somehow, I think that's the genus, but I may be wrong. Okay, so anyhow, you got this subterminal mouth with fleshy lips, and you do business sort of focused on the bottom and the substrate and stirring things around and looking for something edible. And the fish that do that are really good at doing it. You know, they're successful like these others. I once had a little river carp sucker in an aquarium, and it was fun to watch that thing feed. You know, it was a little guy, a couple of inches long, and it would ignore the food as it was falling through the water column and waited till it got on the bottom. And it liked to find a place in the aquarium that was free of sand, just the glass. And it would sort of go around like a little vacuum cleaner and sort of suck up the particles of this pelleted feed or flake feed that I was feeding it. And you could see it sort of milling things and separating, throwing the debris out through its opercles and, I guess, keeping the good stuff. It was a pretty successful little guy. Grew pretty well in that tank. You know, the typical case, though, is that terminal mouth. A bluegill or, lots of fish have that terminal mouth, indicating sort of a tendency to feed midwater. But they're generalists. They can go up, go down. So you can tell a lot, though, by looking at the anatomy of the head, maybe more by looking at anatomy of the internal anatomy, gut, and that sort of thing. And I'll say some things about, and teeth. So I want to, just to have some sort of framework to talk from, I'm sort of going to categorize fishes or feeding habits of fish into what I call major feeding types. And I always ask this on the exam, so make a note there, Chris, in your handout that, Dr. Neal's going to ask us to name the feeding types and characterize each one. Probably I will. And the first of these that I'd like to talk about are predators. And, predators are relatively larger animals, typically larger fish in the community, and they have some common adaptations that allow them to feed on relatively large prey items, usually eating them pretty much intact, without chewing much. They have big teeth, typically grasping teeth, and they have large mouths so that they can engulf large prey items. They have well-defined stomachs with strong acid secretions to digest meat, protein. And typically, because they're eating a lot of protein, a lot of meat, and less of other things, they have relatively short intestines, short guts. The size of the prey relative to the size of the predator varies greatly. But I would say, if I had to, that if you consider all age classes of predatory fish, you're talking usually something that's between 2 and 10% of the size of the predator in terms of prey. We once took a lot of prey out of little red drum, little ones being a couple of grams, and it was almost, you could just about draw the line at 10%. They would eat prey that were 10% as big as they were, and no bigger. A lot of folks like to categorize predatory fish, piscivores, fish that eat other fish, carnivores that eat fish, or piscivores, Piscivore, as either being a stalk and chase type predator or an ambush predator. Largemouth bass is probably a pretty good example of a stalk and chase predator. Marlin is a good stalk and chase predator. They're moving around looking for something all the time, and when they see it, they dart at it and catch it. Not to say they wouldn't lie in wait occasionally, as everybody knows who's fishes for largemouth bass. They like to hide out under the dock or in the brush pile or wherever, and then dash out and grab the prey when it gets close. But there are other predators that really specialize in the ambush mode of feeding. A lot of these are cryptically, or they're marked or shaped in ways that make them hard to see. A lot of them are benthic. I'm thinking now about things like deep sea anglers and sea robins and sculpins and flatfishes like plaice that actually will almost cover themselves up in the substrate and then wait for something to come by and grab it. I guess, and I'm trying to think of, I don't want to get ahead of my notes here and steal all my own thunder, but I guess I'm going to go ahead and say that, ambush type predators, I guess a really great example is the deep sea angler, which has a first dorsal spine modified as a lure, and it's even luminous in the deep sea forms, and it just fishes. You can see on film this lure actually moving, jiggling. It's got a jigging action, and it's designed obviously to attract an unsuspecting prey fish within reach. Telling these stories from words anymore is kind of dull, because all of you grew up watching Discovery Channel, watching nature, on TV. You saw all this, so it's kind of passé now to repeat these things. But I guess I'm going to have to say some things at least to give some structure here. Predators use all their senses to detect prey. Vision probably being the first thing that comes to mind. In turbid water, vision's less important maybe than some of these other senses like smell and taste, which sort of work together in the case of fish. Touch is important and involves the lateral line sense. actually detecting vibrations produced by the movements of prey, the reflection of those. Some fishes have an electric sense. They actually detect. In some cases, they generate a field that is disturbed or distorted by the prey. In other cases, they're actually detecting the electrical activity associated with muscle contraction in the prey. And I remember a fellow named Cal Mean that did some really elegant experiments with some elasmobranch skate. These things, were shown to be able to find the source of an electrical signal that was very, very weak. And he pretty much proved to my satisfaction that they were using the electrical field generated by the prey as a location. They would locate the prey based on that. They would find things that were buried in the sand, little electromagnets. So it's pretty neat to consider all the, we'll talk a lot about, or not a lot, but we'll say some other things, or I'll say some other things and ask you to listen to me about electric fishes and their use of electricity both to detect as well as to actually stun prey. And I'm tempted to look at my notes, but I don't want to. I'm just going to stick to what I can remember and what's on the screen. And what's on the screen is basically, what you need to be aware of. So you need to know what I'm saying too, but the older I get, the less confidence I have in what I say. Now I'm more confident in what I wrote back a little while ago and reviewed again. You ever get that way? You will when you get older. So, that's predators. Eat big bites, big objects, swallow them whole pretty much. Think about a marlin going after a tuna or a largemouth bass going after a carp, hopefully. Grazers. That's a bigger category probably of fishes than predators. It's a broader category, more diverse. These are fishes that feed on relatively small prey items, typically, though, taking still individual bites, seizing individual prey. And the great majority of fishes, including the young of a lot of these things that we think of as predators, fit in the grazer category, and there's typically a transition in life history between the grazing habit and the preying habit in little largemouth bass. Depends on the habitat and how rich it is in terms of forage. But the shift occurs typically somewhere between 60 and 100 millimeters total length in little bass. They quit being little grazers and they start being little predators. Two subtypes I'd like to point out: true grazers, the ones that actually graze almost like a cow. Typically on the substrate, biting off things, biting off pieces of plants or coral, and milling those pieces of coral and extracting the polyps the way certain parrotfishes do, or eating hydrilla, hopefully, if they're a grass carp, and grazing like a herbivorous animal. They are herbivorous. But then there are the midwater grazers that you might think of as sippers. They're not really substrate associated. What they're doing is going around looking for things to eat and opening their mouths, creating a negative pressure field inside and sucking in a little water with the prey. And if you watch a little bluegill in an aquarium, where you can really see it closely and watch how they're eating, they actually don't swim up and surround the prey item so much as they sit there and suck it in. And some fishes are really specialized to do this. You know, they have anatomy that's modified, like pipefishes and seahorses, little tubes that they actually sip through. So they're real sippers. Third category, strainers. Okay, so strainers feed on little bitty things, dense distributions of small prey by filtering it from the water. And one of the common adaptations of these fishes is that they have basically sieves made, you know, from their gill rakers. They have close-set, elongate gill rakers, and they take water in and they expel the water through their gill rakers, retaining whatever is to be eaten from that material. Some fishes are obligatory strainers. Most of the herrings we think of as being obligatory. They don't eat unless they're straining something. You know, they're swimming around as a school and opening their mouths and letting the water pass through their buccal cavities and go out their opercles, and whatever's in it they're keeping. And they're not targeting individual prey. They're just, you know, swimming more or less blindly. Then there are facultative strainers. That facultative implies that you do it when it's to your advantage, but not when it's not to your advantage, as opposed to obligatory, which means you got to do it. Everybody knows those words, right? Obligatory and facultative. And tunas are good facultative strainers. In fact, tunas sort of span the spectrum. One individual tuna can go all the way from predator to grazer to strainer, depending on the kind and distribution of prey. When the prey tend to be smaller and more densely distributed, then the money is on filtering or straining. So tunas turn into strainers when they're eating little stuff. And then as things get bigger and further apart, it becomes advantageous to go after and seize individual prey items and eat those. So facultative strainers tend to shift from grazing to straining as food item size goes down, gets smaller and smaller, and the density of the individual items go up. Number 4. So what have we talked about? Predators. We've talked about grazers, 2 different kinds of each, I guess. And strainers, or filterers if you prefer. I'm not a stickler for the specific word, and if you can't remember on the exam that I said strainer and you say filterer, I'll probably say fine. I'm not going to hammer you for that. It's the idea, the functional basis of the idea that's important, and not so much the labels that we use. Although, in order to fit in, with your professional crowd, you got to know the buzzwords. It's always buzzword bingo, right? You got to be able to say the words that everybody is going to say, oh yeah, they know what they're talking about. But I would say you need to know the ideas rather than the words, and that's more important. So suckers, some fish are really suckers. And the Catastomidae, I've mentioned already, the North American sucker family is the classic example of suckers. But you could think of some others. Sturgeons make pretty good suckers too. They suck food off the substrate, literally, like a vacuum cleaner nearly, except a water version of that. And the common adaptation, as I've already shown you, is a subterminal mouth. And often these fish have fleshy lips, and the lips tend to be very sensitive to touch. Some of them have got barbels that are sensitive to touch and taste. But I don't think the catastomid suckers have any barbels. I'm almost sure that I'm right about that. The buffalo is is an example of a big catastomid buffalo fish. Does everybody know buffalo fish? And I don't think they got any barbels, but carp, are they look similar superficially to a buffalo, but they have barbels. I don't think goldfish have barbels, right? But little carp, have barbels. Anyhow, they got some head-in stuff that's sensitive to touch and taste, typically, and they use it. Finally, we get to parasites. My parasitology teacher 50 years ago used to say that a parasite is just a really sharp animal that's lost all that worthless equipment it didn't need. It depends a lot of time on your equipment. It doesn't have to have a lot of digestive equipment because you already done the work, maybe. And a really good example of this is Serratius, the angler. In the deep sea forms, I like to tell this story. I've been telling it for a long time. You've probably seen this story on the Discovery Channel already about the little dwarf male that's parasitic on the female. The male just degenerates to an animated gonad, basically, and the female develops this little papilla, and the male attaches and sits there and takes this nourishment from the female. That's the ideal parasite in a lot of ways, and the advantage to the species, of course, in this case is that make sure the two sexes are together at spawning time in a pretty rarefied world, where it's few and far between other anglerfish. So, Serratius, that's kind of an interesting fish. It's kind of a fine line between parasitism and predation or grazing. A lot of fish like Gambusia, for example, the Piscileids in general, are bad fin nibblers. Keep them in a tank with their fish, they're always nibbling on their fins, they're eating them up. Well, that's not really parasitism, I guess. That's sort of grazing. They're not killing the host, but then most parasites that are really successful don't kill their host, at least not right away. They take their time at it. I'm thinking about lampreys. Those are pretty good parasites. They're finding a nice lake trout and latching on and rasping a hole with that radula, and fastening on and then drawing the body fluids out. Hagfish are even more vile. They tend to be more scavengers than parasites, although they live on living hosts, but they will happily eat a dead host, and they make a hole and they go right inside and sort of wander around eating as they go. Trichomecterids, the little parasitic catfish family from South America. People have heard about this and probably seen it on the Discovery Channel. These things are parasitic naturally on the gills of other fish. And fishes produce most of their nitrogenous waste by way of the gills. And so these things are attracted to urea, maybe to ammonia, maybe to certain amines as well. And in the case of humans, of course, we produce our urea at a different site, and that's where these nasty little fish go. They will penetrate so far up the urethra, both male and female victims, that they have to be removed surgically. It's hard to know what is the cure as bad as the attack in the first place, or I don't know. Probably not. Probably the cure is not as bad. Throw out their little pelvic spines and pectoral spines and lock themselves in place. So I understand, and I get my information like you do from the Discovery Channel, that the people that are familiar with the situation, both men and women, use certain prophylactic measures when they get in the water with these guys to avoid this kind of attack. So that's the trip through the diversity of fishes, and it's a weak effort. I realize that. You could think of another 100 examples of really interesting cases and details But I think that's the tip. That's the picture. You know, the game is to gain nutrients and nutrition from other critters. Dr. Weinmiller will spend a lot of time talking about food webs and pyramids and chains and who eats whom. Trophic relationships, trophic levels. So the shorthand that folks tend to use is P for producer and C for consumer, and a 1-2 subscript to indicate the level in the trophic system. So a first order producer is P1 actually. I guess there's only one level of producer, and that's eaten by a consumer that's an herbivore. You know, it's eating something that is normally a plant, at least an autotroph, something that's, you know, fixing carbon. And that first order consumer then is consumed by a second order and a third order and so forth. So we're going up or down depending on your perspective, the food web or the chain. Sometimes we talk about pyramids to emphasize the fact that the bottom tends to be broader in terms of both the diversity of organisms as well as the biomass than the peak. So predators are at the apex and all these producers are down at the bottom making the foundation of the pyramid. There's a rule, and I don't think it's been negated, probably challenged lots of times and lots of variation, but the rule is 10% of the energy at each trophic level is captured by the trophic level above it, and 90% is wasted. That's the 10% ecological efficiency rule. You know, rules are meant to be broken and always stimulate the search for exceptions. But still, it's important to have some kind of central rule in mind when you start looking for an exception. And that's the rule I learned. You probably did too somewhere, freshman or junior college or wherever you did it. So there's some examples, you know, just to show you the kinds of diagrams that people make. Over here on the right is a chain, which is a simpler system with, you know, just a linear arrangement of producer and consumer. And then over on the left is something to emphasize the pyramid idea, where you got 3 levels of producers, or 3 different kinds of producers, and consumers at the first level, which would be the herbivores, are eating the producers. And it looks like C11 here is a little more of a generalist than C12, because C12 is only eating P2 and P3. It's not eating P1 for whatever reason. You know, these are the ways that you assess these trophic connections. And then up here we've got a carnivore, which is eating both of the primary consumers. So there's that 10% rule, and it's kind of interesting to consider the algebra, or the geometry, I guess it really is, of the situation. I mean, by my scheme here, it takes a metric ton of diatoms to produce 1 gram of bluegill? Probably not. You know, that would be a really extreme food chain. But the logic is that it's a 10% transfer, right? Diatoms are eaten by rotifers, which are eaten by cladocerans, which are eaten by copepods and minnows and dragonfly nymphs and bluegills. And one of the points I want to make here is that it's not always a vertebrate eating an invertebrate. You know, it can be the other way around. Dragonfly nymphs do eat minnows. These 3 here are zooplankton groups, and down here are phytoplankton, diatoms. And of course, it's easy to think of short circuits in this kind of system. You know, you could have, for example, diatoms being eaten. Well, I wouldn't want to go too far. Maybe diatoms being eaten by cladocerans. And certainly bluegills can eat copepods, you know, so it could be a couple or 3 steps instead of however many I got there. And if you shorten the chain, then presumably a greater proportion of production at the bottom reaches the top. And this is something every farmer knows and rancher, you hope, and every fish farmer, so you go out and you grow tilapia, let's say, cichlids, and these things are, in some cases, herbivores. And so maybe you get a lot, for per unit production, you get a lot of tilapia meat out of the deal. That's the goal. We always try to shorten these food chains, even in systems where we're growing things that are higher up, like red drum. But there's always a risk, too, and the risk is when you give up the redundancy in the system, you lose the safeguard of being able to find alternate pathways through the system. And so one of the risks of efficient agricultural production is that you have a fairly stable system, a fairly unstable system, a system that's prone to break down. If anything goes wrong, everything goes wrong. So you have to really manage those kinds of systems very carefully in order to keep things going and avoid this kind of left turn that leads to disaster, an accident. Everybody knows that, though, right? So the more diverse the system, presumably the more stable the system. And there's been a whole lot of ecology done along those lines to document, to support, to test the idea that a diverse system is a stable system. I'm going to talk a little bit now about not what fish eat, but how much they eat, the intensity of feeding, as V.S. Ivlev, the Russian who did some of the classic pioneering work, called it. Ivlev, I-V-E-L-E-V. I probably got his name in here someplace else. Anybody wants to see it? I've got, one of probably only a few copies in Texas of his little classic monograph called The Ecology of Feeding Fishes. Anyhow, there are these intrinsic factors, things that originate inside the fish, but intrinsic is sort of with quotes, I guess, because everything originates outside the fish ultimately. But in a proximal sense, the origin is internal. And it has to do with this idea of hunger and satiety or satiation. And, how hungry are you? Well, the way Ivlev would define hunger is you're as hungry as the amount you eat. In other words, that's the test. That's an operational definition of hunger, as opposed to a sort of touchy-feely definition that says, well, I kind of feel hungry and I kind of crave this and, I'm really thinking about food. Well, all of those things are true, but hard to measure. And so if you're dealing with a fish, he's not talking much, typically, so he or she. So you got to, ask the question in a way they can answer it. And one way to ask the question is lay out the food and measure how much they consume. That's how hungry they are. That's an operational definition. Food deprivation, typically increases hunger, measured that sort of way, but not indefinitely. Sometimes deprivation over a period of days or more can result in, a dysfunctional system that refuses to be hungry. Extrinsic factors, abiotic and biotic. That's the two categories, I guess, that are possible. Abiotic being the non-living components of environment, and biotic being the living. And I want to be careful not to be redundant over labs. Some of this material I've already reached in and got. We don't have to cover it again. I'll ask you to review that lab presentation that I gave background for Lab 1 in the context of all of this if you need to. Light is really important, particularly for visual feeding fishes. And often such light-sensitive feeders tend to show dawn and dusk peaks. We use the word crepuscular, C-R-E-P-U-S, C-U-L-A-R. I'm trying to see if I've got the word in here anywhere. Probably don't. Crepuscular animals are animals that tend to be most active at the interfaces between night and day, both dawn and dusk. And there are lots of reasons for that kind of behavior. Yeah, I got it. There's the crepuscular. One is that you want to be active in feeding when your prey is active, and if you're a diurnally active predator feeding on a nocturnal prey, then you better, find a time near dawn or dusk to get at it. So you got to have an overlap between the activity of the prey and enough light to see the prey. And one of the best examples I know of that kind of relationship was one researched by a colleague of mine. And what he worked on was yellow perch feeding on the amphipod hylela. Do I have a slide in here about that? Yeah, I do. I'll get to that. So let me say before I get to that slide that we got this activity overlap with the detection ability on the part of the predator. And then the second thing is maybe a derived relationship having to do with the fact that you got a food processing rate that enters into it, and there's evolutionary adaptation to the day-night interval, so that if you fill up, at dawn or an hour or two later, then you digest that food and process that food so that you're hungry again by dusk. It wouldn't be smart to not get hungry until after dark and your prey is no longer available. So you'd have to wait till the next day then to eat. So you want to time your physiology to match up with the diel cycle of the environment. So there's a certain amount of that going on, and in tunas that looks to be really important. I've got something to say here about visual stimuli and feeding releasers, but first I think I want to say something about Bob Mullins. That's the guy that did this work, my colleague back in graduate school, yellow perch feeding on hylela. And so what we've got is that, the more light there is, the better you can see the hylela. But hylela tend to stop being zooplankton and start being attached herbivores grazing on aquatic plants when the sun gets up. So what happens is that you've got this visibility of hylela to yellow perch that's maximum during the daylight period, but you've got availability of hylela to yellow perch high at night and low during the daytime, because during that period they become virtually invisible because they are sticking to the vallisneria and the oscillatoria. Not oscillatoria, that's not the word I'm looking for. What am I looking for? Vallisneria would be good. That's what, eelgrass, I guess. What's coontail? Or somebody doesn't know the plants here, and I'm one of them. Anyhow, so during the daytime, hylela live in the littoral zone associated closely with aquatic vegetation. They become almost invisible to yellow perch. But then when the sun goes down, hylela turn loose of the plants and start to become zooplankton and actually migrate out of the littoral zone, the weeded, vegetated shallow water of a lake, and into the open water or limnetic zone. And it's during that transition, when they're visible and active, that they get really nailed by yellow perch. And so you can do these neat little feeding experiments where you got captive yellow perch in cages, and you sample them to make sure when they're feeding, and you're measuring and that's what Bob did. Really worked out neatly. So here's something a little more current and a little more optimistic. Particularly this time of year, we're coming into the season when red drum form large schools, of fishes in the passes, like here at Surfside. And they're feeding and they're spawning, and boy, they're red. So you can see how red they are. And if you look carefully, these fish are going after something. You can actually, I think maybe my next slide. Do I have another one? I'm trying to see if. Yeah, I think there's a menhaden there, huh? He's nailing some. They're nailing menhaden, Gulf menhaden. So it's easy to see why they're red fish. There's other red fish in the world, but we call red drum red fish too because they're pretty red. So that's a happy sight to conservationists and to fishermen, fisherwomen, fishers. And, because 30 years ago, you couldn't see that. 30 years ago, red drum were far and few between. And due to a combination of management of limits and banning of commercial fishing and banning of entanglement nets and stocking of little juveniles from hatcheries, that is one of the great success stories, I think, in Texas fisheries, is the return of the redfish. And this site now is to be seen where it couldn't have been seen when I came to Texas, because that was just about the time, 1970s, when red drum were at the minimum. They were never endangered, but they were very scarce, overfished. So that's one of the other fish that I've worked on. Ray Kemps will know that's my hand-drawn tuna. That's a skipjack tuna. And skipjack are pelagic, fast-swimming fishes. Tuna, skipjack are among the smaller tunas. Probably 20 kilograms, maybe almost world-class skipjack. Probably most of the skipjack that people would see would be in the 5 to 10 kilogram range. Katsuanus pelamis is the species. I guess it's still Katsuanus pelamis. And these are fish of the Central Pacific. And when they're just cruising around, they got this kind of color pattern with these iridescent purple. That's a very common color in pelagic animals, is this sort of a violet purple, beautiful color. They're counter shaded. They've got dark above and they got ventral surfaces that are silvery. And the whole idea is that if you look down on them from above, they're hard to see against the dark bottom or the dark deep water, which itself can look almost purple when it's clear. And if you look up on them, then you're looking at those silver venters, and against the sky, it's hard to see them. So anyhow, they are really good predators, and they'll cruise around in a school of a few thousand to maybe 100,000 individuals. A school that may be 4 or 5 kilometers wide, and they're looking for something to eat all the time. And when one edge of the school finds something, rather than being greedy and selfish, they share. And they share, instinctively. They probably would rather not individually if they could avoid it, but the species has learned that it's more successful to do it that way. And those fish that find something to eat develop feeding bars, which are vertical bars, very obvious to humans and to other tuna, presumably. And that feeding pattern propagates from the fish that found the food across the school at a very rapid rate. And pretty soon everybody veers and the whole school heads toward the source of food. And we worked on tunas at Koala Basin when I was with the Fisheries Service in Honolulu, and we were easily able to generate this response in captive fish. And one of the neat experiments that we did was we had a cutout of a tuna on a piece of plywood with a broom handle stuck in it. And one side of that cutout was painted with this pattern, the non-feeding pattern, and the other side with this pattern. And the school of fish would go cruising by and you'd present them the cutout with the horizontal streaks, the non-feeding pattern. They would just swim by. You know, you can see their big, smart eyes. They look smart. I'm not sure they are, but they got great big eyes, you know, and they're very visual. And their optic lobes are big. And they'd look that over and just grown by. Then you flip that thing around and show them the other side. And, oh, they do figure eights and come to a halt. You know, it's almost like a cartoon response where, you know, put on the brakes. And they don't really have good brakes, but they can turn. And then they would go into a figure eight search pattern. And they themselves then would develop the feeding bars, even though there wasn't any food there. You know, they had seen a fish that said, hey, I've detected something good. So that was pretty neat. We used that to train them. You know, we could cause fish swimming in a donut-shaped tank to reverse direction by presenting them one side of a fish or another. We could make them turn. And we took advantage of this inherent instinctive response to go into a search pattern when they see the feeding bars. So basically we could say, hey, swim clockwise, and they'd go screaming around the tank clockwise, turn it and say, swim counterclockwise. There's a tuna hook. I used to have the real thing. I think I took it home, though. That one's probably for a 200-pound yellowfin. That's not one for a skipjack. But notice there's no barb on that dude. You know, and in fact, you don't need feathers or anything else. Bait, no squid. Just the brass and the hook is all you need. Once these fish go into this frenzy feeding mode, anything that goes in the water is food. You know, the prey fish that they're looking for, cigarette butts that the crew has washed over the side. Nobody smokes anymore, but they did back in those days. Or one of these hooks. And they'll take it, you know, and the reason there's no barb is because it's not because you don't want to hurt the little tuna. It's because you want to catch as many tuna as possible. So the motion is the hook goes in the water, the fish hits it, immediately it's jerked out of the water, slung over the fisherman's shoulder, onto the deck, a quick shake, back in the water. How fast? This is a number that's hard to believe, and people always look at me like, but it's been timed. 5 fish per man second. I don't know how you can do that. That's what they actually record. Maybe there's doubles or something there. I don't know. So, you know, these big tunas, the 200 and 300 pound tunas, it takes 2 guys or 2 fisher people and their guys to catch one of these. And so you have a bridle, you know, two heavy, stiff bamboo poles and heavy lines coming down to a Y and then down to a single hook. And 2 people fish together, maybe even 3 people in some cases. I'm thinking back what I said. I think it's 2 fish per man second. I wouldn't want to overstate it. 2, not 5. 2 fish per man second. I'm pretty sure that number is a good number. There's a machine that was developed by the Japanese called the rowback fishing machine. And what it is a robot that does this. You know, it's really literally a robot that holds a cane pole that is just like the one that the fishers, the live human fishers use, and has a tensiometer associated so it detects the jerk and jerks itself and swings the fish, and those things can outfish humans. I don't know if they're still using. These stories are 40 years old now, nearly. And that hook's older than that, probably. So there's a different kind of hook. So that's a little more recent vintage, actually, but it looks a little cooler, right? That's a cod jigger. That's a fish used to catch Atlantic cod. And that one came from Newfoundland. And back here in the back end there in the caudal fin is the hole where the line is fastened and the thing is jigged head down. And it's done a lot of damage to cod. So much so that for a while there weren't any cod around to be caught, but I think cod may be coming back. Factors affecting the intensity of feeding now: abiotic, extrinsic factors, continued temperature. I was talking really that whole foray there was having to do with light and vision, and I probably went further afield than I needed to, but you found it entertaining, right? So that's good. Going away from light and talking now about temperature, feeding rates tend to go up with increasing temperature, in keeping with the fact that you better have more to eat if the temperature is higher, because your metabolic processes are running faster, you need more food. And that continues up to a point characteristically about 2 degrees or so below the upper lethal. And at that point, fish go off feed and won't eat. And a change in either direction, as every fisher person knows, abrupt changes in temperature will put fish off feed, so they don't want to bite so much if there's a front coming through. Photoperiod, the length of the light part of the day between sunup and sundown. Increasing day length tends to trigger increasing feeding rates in fish. Longer days, they eat more. This maybe is sort of anticipating a high temperature period, fast growing season, so you want to really stuff yourself if you can in the spring when the day length is increasing. But it turns out, I should say, not but, that this photoperiod effect on feeding intensity tends to be independent of temperature. And the classic experiments were done by a Canadian researcher named Margaret Brown, and what she did was keep trout in tanks and show that even if you keep them at constant temperature, they feed more as photoperiod is increasing. So when temperature is maintained constant, the fish are still responding, and they're responding to photoperiod rather than temperature. In addition to temperature, but in this case, she separated the effects. And I think lots of other people have done similar experiments. DO, I think I mentioned this in the lab front end, that DO limits feeding and food processing, and looks like the level is around 5 parts per million, and that seems to be pretty much independent of everything, which is surprising. It's 5 parts per million in sockeye salmon. It's also 5 parts per million in red drum. And I've talked to you about the DO limitation on food processing rate, and there may be an accompanying depression of appetite. We talked about that too. I think there's less assurance about the appetite part. So that's the graph. I mean, it's almost that regular. Here's a percentage of maximum consumption as a function of DO, and it's pretty much 100% at high DOs, and when you hit 5 or so, it goes south. And by the time the DO is down to 2 or 1, consumption virtually goes to 0. That's long term, where you're feeding fish, maintaining them at those DOs, and so it's hard to know whether the effect is on appetite or food processing, because fish can't fill its stomach with any more food if it's still got food there from yesterday. Biotic extrinsic factors. This brings us to concerns about the kind of food and its availability. And food distribution, if you got just one kind of food, Ivlev, what do I want to say about this? Well, I guess what I want to say is if you got one kind of food, then we talk about feeding intensity as a function of food availability or food density, and gradient responses to the variation in availability. If you got more than one kind of food, that leads us into a consideration of feeding electivity or selectivity, electivity indices. And I think what I had here in mind was just to start. And I'm looking at the clock and I'm thinking that, I'm going to wait till next time to finish this because it's 12:22. I don't want to get in the habit of overstaying my welcome with you and having you be late to your next class, or either one of us to be embarrassed by you walking out on me. So we're going to stop there for today and pick it up on Thursday. Again, lab this afternoon, help session locally. I hope people who are at a distance are doing their lab work also. I'm going to have a few words to say at the beginning of lab about downloading protected files using the password that I gave you. So you might want to come for that even if you don't plan to stay the whole time. All right, we'll stop there. 2 fish per man-second, right? Not 1, not 5, not 50, 2. Still, that's a lot, though, right? Half second per fish.